Hello students, I'm Dr. Arpita De. A very warm welcome to my YouTube channel. The topic for today's discussion is ultrastructure of cytoskeleton. Before we delve into the topic, let's first see what is cytoskeleton, what is meant by cytoskeleton. The cytoskeleton is the skeletal framework of the body. What is skeletal framework? What does skeleton do in your body? The skeleton gives you a proper shape. It gives you strength. It gives you the power to resist mechanical force. Same thing applies to cytoskeleton. It gives proper shape to the cell and gives it the power to resist deformation. So it's a complex dynamic network of interlinking protein filaments present in the cytoplasm of all cells. In eukaryotes, it is composed of three main components: microfilaments, abbreviated as MF, intermediate filaments, abbreviated as IF, and microtubules, abbreviated as MT. A multitude of functions can be performed by the cytoskeleton. Its primary function is to give the cell its shape and mechanical resistance to deformation. It associates with extracellular connective tissue and other cells to stabilize the entire tissue. Hereafter, we will use these abbreviated terms MF for microfilament, IF for intermediate filaments, and MT for microtubules. These are the three components that make cytoskeleton. The microfilament is approximately 7 nanometer in diameter and is known to be the narrowest among the three. The intermediate filament has a diameter of approximately 10 nanometer and microtubule has got a diameter of approximately 25 nanometer. The microfilament is the narrowest of all and they are associated in a parallel manner. The basic building blocks present here is actin. These actins are abundant intracellular proteins and they exist as globular monomer, which is known as G-actin, and filamentous polymer, which is known as F-actin. The microfilament have two structurally different ends. Why do we call it structurally different ends? Because the polarity is different. One end is the slow-growing or minus end, also known as the pointed end, and the other is the faster-growing plus end or barbed end. The basic building block of microfilament is actin, as we have just now mentioned. It is an abundant intracellular protein, and the actin isoforms can be classified into three groups: alpha-actin, beta-actin, and gamma-actin. Now let's see how does growth take place in this filament. The polymerization of G-actin can be divided into three phases: nucleation, elongation, and steady state phase. In nucleation phase, we see G-actin subunits combine to form oligomeric subunits. This is basically a lag phase where G-actin subunits combine into 2 or 3 subunits. When these oligomeric subunits reach 3 subunits length, it acts as seed or nucleus for the next phase. The next phase is elongation. From the word elongation, we can understand that the length increases. Growth of F-actin filament through addition of G-actin to both its ends. As F-actin filament grows, the concentration of G-actin monomer decreases until equilibrium is reached. And finally, we come to the steady phase where we see no net change in total length of the filament. G-actin monomer exchange with subunits at the filament ends, but there is no net change in the total length. F-actin grow faster at the positive end than negative end. The rate of addition depends on free G-actin concentration. The rate of addition at the positive end is 12 per micrometer per second and 1.3 per micrometer per second at the negative end. The rate of dissociation does not depend on the concentration of free ATP G-actin. Subunits will be added at high free concentration of ATP G-actin, but as the concentration is lowered, a point will be reached at which the rate of addition is balanced by the rate of loss and no net growth occurs at that end. This is called the critical concentration. Now let's carefully look into this picture. This is a diagrammatic representation of polymerization of the filament. Here we see G-actin without ATP. And these are ATP molecules. This G-actin will combine with these ATP molecules, and after combining with ATP molecules, it will get assembled at the plus end or growing end. When these G-actin molecules are without ATP, that is ADP bound actin and PI, phosphate, here disassembly is seen to occur. This is the minus end event. Now coming to actin treadmilling. What is actin treadmilling? At steady state, when free ADP G-actin will be intermediate between critical concentration of plus and that of minus end, then the plus end grows and minus end lose subunits. What is the actual meaning of the statement? It says that at steady state, when free G-actin bound ATP will be intermediate between the critical concentration at the growing end and the critical concentration at the minus end values, then the positive end will grow. This means when will the positive end grow? The positive end will only grow when this ATP bound G-actin will be intermediate between the two values. Which two values? The critical concentration value at the plus end and critical concentration value at the minus end. This is called actin treadmilling. This process is mediated by two proteins, profilin and cofilin. Now we will come to motor proteins. This is something very important and interesting as well. What is motor protein? Motor proteins are proteins that generate force required to propel the movement of molecules within a cell. They convert chemical energy stored in ATP into mechanical energy or work, which is used to move cellular cargo attached to the motor. Motor proteins can be grouped into three broad families: myosin, kinesin, and dyneins. First, we will talk about myosin. This actin filament serves as tracks for myosin motor protein movement. Myosin is made of head, neck, and tail domain. The head domain interacts with the filamentous actin. The neck domain serves as a linker and the tail domain interacts with cargo molecule. I've drawn a very simple diagram to depict these parts. Please try to tally it with the write-up in this box. Okay. Now we will come to intermediate filaments. Intermediate means what? From the word itself, we can understand that it lies somewhere between the microfilament and the microtubules. This is the diagrammatic representation of intermediate filaments. It is stable, unpolarized filaments with great tensile strength. Why do we call unpolarized? Here we see there is no plus end and minus end as we had seen in the previous case. Here we have an N-terminal region and a C-terminal region. The entire filament can be divided into three parts: the head, the coil region known as the rod, and the tail region. IF or intermediate filaments can be classified into four types: the keratins, the desmin and vimentin, neurofilaments, and lamins. These are the four types of intermediate filaments that are found. They are extremely stable. The primary building block of IF is a dimer. The dimers from tetramer, where the two dimers are in opposite direction. The tetramers assemble end to end and interlock to form long protofilament. Four protofilament associate into a protofibril and four protofibril associate side by side to form a 10 nanometer intermediate filament. I'm leaving a question for you to answer. Can you say how many protofilaments are present in this intermediate filament? Please write your answer in the comment section and let me know the calculation, the result of your calculation. Now we will finally come to microtubule. What is microtubule? It is the major structural component of cytoskeleton. The building block of microtubule is tubulin. Tubulin is the major structural component of MT. It occurs as a dimer of alpha and beta subunits. Tubulin subunits associate with each other to form a protofilament. Lateral association of 13 protofilaments, please note the number 13 protofilaments, makes an MT. Each tubulin subunit binds with two molecules of GTP. Both subunits bind GTP, but GTP associated with alpha subunit never dissociate, but that associated with beta subunit dissociates. Please keep this in mind. This is the picture. One is the alpha subunit, the other is the beta subunit. This is the tubulin heterodimer. This is the protofilament. If you see the cross section through the MT, this type of a structure will be seen. Please count the number of subunits. You will see 13 subunits in the cross section view. This is something very, very important as well as interesting. What is MT dynamics or microtubule dynamics? Microtubule can oscillate between growing and shortening phases. This dynamic behavior permits a cell to quickly assemble or disassemble microtubule structures. MTs assemble by polymerization of alpha-beta tubulin dimers. By now, you have definitely understood what is growing and what is shortening. Growing means what? Subunits will add to the growing end or plus end, and shortening means what? Disassembly of subunits will take place, which will cause shrinkage of the filament. Now, two words here will be very, very frequently used. One is rescue, the other is catastrophe. What is rescue? Rescue is the association of GTP beta tubulin at the end of each protofilament causing growth of the filament. The growing MT has the GTP cap. Now what is catastrophe? Catastrophe is that phenomenon where rapid disassembly of GDP beta tubulin at the end of each protofilament occurs. This causes shrinkage of the filament. The shrinking MT doesn't have the GTP cap. Now let's read this carefully. MTs are assembled from heterodimers of a GTP bound forms of alpha tubulin and beta tubulin proteins. Unidirectional orientation of tubulin heterodimers in empties result in polarized nature of empty. Obviously, polarized nature comes from the two different polarity that are there in the filament. One is the plus end, the other is the minus end. Rather, one is the growing end, the other is the disassembly point or disassembly end. So, unidirectional orientation of tubulin heterodimers in empties results in polarized nature of empties with alpha tubulin exposed ends or being relatively stable ones and beta tubulin exposed ends being more stable. Upon empty assembly, most of tubulin-bound GTP, with exception of subunits at the very ends of empty, is hydrolyzed in GTP. The energy released by the hydrolysis of GTP to GDP results in decreased stability of empties and leads to growing and shortening phase transition at the empty ends called dynamic instability. In addition, at the polymer mass steady state, the growing and shortening transitions at the opposite end of empty gives rise to net shortening. This directional growth of empties is called treadmilling or flux. Now, finally, we'll come to the empty-based motor proteins. There are two empty-based motor proteins. One is the kinesin, the other is dynein. Kinesin is the plus end directed protein involved in transport of organelles and vesicles, and dynein is the negative end directed protein involved in transport of organelles and centrosome assembly. So, kinesin will carry the protein towards the plus end and dynein will carry the protein towards the minus end. This diagrammatic representation shows the direction of cargo movement with the help of dynein and kinesin. This is how we come to the end of our lecture. If you have any questions, doubts, please feel free to write your doubts and questions in the comment section. I will try my best to get back to you to clear your problems. If you like the video, do hit the like button. This will act as a motivation for me, and do subscribe to my channel to get updated about my new uploads. Thank you so much for your kind attention.